Awesome. Well, I just started recording, yeah, so thanks for coming back this week. And I take it this week we're going to dig into R a little bit more and some data. So, and also Gina will post. We did not yet post the one from last week. So if any of you who are here didn't see last week's, that kind of gives you the big overview, we will post the link to that as well soon, and we'll share it with you all at Neon too. But yeah, so take it away.
Great, yeah, thank you so much. We're so happy to be back here. We have Arlene back with us again, and but I think Arlene's mostly going to be kind of an observer for today, but I wanted to introduce my colleague Marie Faust, who will be joining us for today. Marie works with me here at headquarters, and I guess maybe I should just let her give an introduction. Thanks.
Hey, everybody. Yeah, so I work with Donald on our education, engagement, and outreach team. My technical title is a science outreach specialist, but I kind of wear a bunch of hats and run some of these workshops with Donald, but also do social media and just kind of various things for communications and outreach for the NEON program. And I work closely with pretty much all of our domains and field sites. So if you have specific questions about the field sites or the domains, please feel free to reach out to me today or through email. And then just a quick little bit about my background. I went to grad school at Northwestern, and I'm from the Midwest, so I'm very familiar kind of with Iowa and kind of the plant communities around there because I'm a plant biologist. And yeah, if you guys have any questions, I'm happy to answer any and all that I can.
Perfect. Well, thank you, Marie. I'm going to share my screen now and pull up our agenda for today. Great. Hopefully folks can see this here. I'll take this link and throw it into the chat window as well. But really what we wanted to do today was to start by clarifying any questions people may have resulting from last week's discussion, or if you're new to us here this week, if you have any kind of overarching questions about the NEON program in general. So we'll be happy to discuss that for the next 5-10 minutes or so. So yeah, let's just open it up at this point to questions. I love having these intimate little groups. So feel free to unmute yourself, throw on your webcam if you want to communicate with us here. So yeah, let's see if there are any outstanding questions here.
I'll jump in. So my image of NEON is spatially extensive at the macro scale, but essentially one site per ecological domain. Do you also have data that has, say, more spatial resolution at each one of your macro sites? So maybe something that's collected on 10, 15, 20, 30 different small areas or not so small areas around your towers?
Yeah, I think that's a great question. Marie, you look like you have an answer.
Yeah, so I just have a quick example. So most of our terrestrial sites actually have sampling at plots around our towers and plots that are not next to our towers. So we do kind of have smaller, I would say more community ecology focused sampling, but you can get it to an even smaller kind of resolution as you may. So a good example is in Rocky Mountain National Park, we have specific plots. I just did really small analysis on elk browsing actually on aspen trees there for the Park Service, but so we have plots, and I wanted to look at different plots at elevations throughout our sites. And the sites with the towers, I was able to look at the tower plots, which are actually enclosed next to the dispersed plots. And so you can see Donald's kind of showing that right now on the screen. And they're not dispersed plots, distributed, excuse me. But it kind of allowed me to do this kind of community analysis. And Donald, I don't know, do you have, I could pull up a ton of other examples, but the quick answer is yes. And Donald, I don't know if you want to bring up any other types of examples.
Yeah, I think your example is a great one. Another one that comes to mind is soil quality. So we have kind of distributed soil sampling regime that occurs around our sites. But Philip, I think you were going by Phil, if I remember last time.
Philip, I
Dr.
respond to everything all the way down to hey you, so don't worry about it.
All right. Well, Professor, let's say, doctor.
That one.
All right. So yes, we have a very intensive spatial sampling regime right next to our tower. So what I did while Marie was talking, I hope it wasn't too distracting, was just pull up one of our site web pages here. This is for the Wind River Experimental Forest in southern Washington state. And so here, this is a nice kind of GIS enabled page. You can pull up satellite imagery or whatever if you want to. But I like to bring this up because I think it gives a good sense of how kind of spatially organized these sites are. So we have our tower, which is the most conspicuous object on the sampling area. And then within the tower, what we call our dominant airshed. So this is, the land across which we have detected that most of the wind comes from for most of the season. Some places have two dominant airsheds because they have really different kind of seasonal wind patterns. So we have a really intensive sampling regime that occurs within that area. So there'll be all types of samples such as vegetation structure, which I'll bring up in our discussion about the coding section here soon. And then outside of the tower plot areas, we have a whole network of distributed plots as well, where we make these observations. What I will say though is that it's still fairly limited in terms of spatial extent. So here at Wind River, and this is common at many of our other sites, there are two dominant sampling boundaries, which are indicated by this black dashed line here. This is basically land upon which we have permission to do our sampling. There is some disruption from people walking around, digging holes, things like that. So outside of these boundaries, we don't have any samples except for our airborne observation platform. So our remote sensing sampling area is a minimum of 10 kilometers by 10 kilometers. The scale bar is shown down here on the bottom. So I think for Wind River, it's actually quite a bit bigger, maybe by like 15 kilometers by 15 kilometers or so. And then we'll have all sorts of spectral information and LiDAR information from that area. But I absolutely agree with you. If you wanted to zoom out more and more and more, it's like hundreds of miles between our different sites. One thing that we do to try to account for that and accommodate for that is, and Marie, maybe you can help me out here if I'm using the wrong terminology, but what we currently call them are our relocatable sites. So we have our core sampling sites, which are going to be in the same location for the 30 years of our observatory. And then our relocatable sites were developed with this exact question in mind. How do we try to capture these massive gradients that occur between our sampling sites? So our relocatables are, they're basically like a tiny home version of our infrastructure. It can fit on the back of a letter. It's basically like a shipping container that we've modified to have all these instruments on it. And so we can move these around and either, one, use them to fill in locations across a gradient. So for example, like from Moab up to Rocky Mountain National Park is a pretty big area and they're really different environments. So we try to sample places in between. We can also use those for kind of temporally opportunistic sampling. So if we have a big wildfire and we want to see what the response is like to that, or I think a good example is like if there's a big hurricane in the southeast, we can deploy one of our relocatable sites to that area to try to get that response to the disturbance and make those observations. So I think you bring up a really important point, and being a trained geographer, I could talk about this forever, and I'd be happy to go into more detail about it, but I'm really glad that you brought it up. Thanks, Phil.
No, well, thank you.
Cool. Marie, do you want to chime in on what are we calling relocatables and what are we doing with them?
So there's relocatable sites, which are actually going to probably stay permanently. And then there's our assignable asset team, where we have these assets like Donald was talking about, where essentially you apply for funding, and we have these mobile deployment platforms that can fit in the back of a very big truck, and you can bring them to the sites to measure all different kinds of things. I did want to point out for all my aquatic ecologists out there that we do sample multiple reaches at sites for all of our like observational products. So, you know, there is some, you know, community kind of like smaller scale sampling at our aquatic sites as well. Just wanted to make sure that got out.
Good point, Marie. Thank you. Okay, are there any other kind of burning questions before we move on to the R coding section, which I know is what people are really here for? All right, well, let's just go ahead and get into it then. On our agenda, you'll see there's this section that says downloading and exploring Neon data in R. I'll take this URL and throw it into the chat window as well. So what this is this is one of our tutorials that Arlene introduced you to last week. Our tutorials, just really briefly, are designed to be kind of use as you please, work at your own pace, totally kind of independent, asynchronous learning materials. These are things that I think a lot of educators adapt to use in their classrooms. If you're curious about that, definitely follow up with myself, Arlene, or I think Marie does a lot of this. This is really one of her specialties, to integrate this information into undergraduate classrooms in particular. So I just wanted to kind of throw that out there, but a quick orientation to what our tutorials look like. We always have a navigation bar on the right side. So these headers will take you to different places in the tutorial. At the top of each tutorial, there is usually a little kind of summary about what we're going to go into. There's this section in a gray box that will tell you which packages that you need to install in R to get this to work for you. And then we have all of these code chunks. So if you've worked in R Markdown documents before, you probably know what this looks like under the hood. But these code chunks are completely copy and pastable. So anything with the black background here, you should be able to highlight, copy, and then paste right into your own console or R script, whatever you're working in. We like to put in a ton of additional links for your own education and just kind of additional information. And then, of course, we'll get into kind of the meat of the tutorial. But before we get started on that, what I want to do is scroll all the way down to the bottom. And at the very bottom of each one of our tutorials, we have this link. It says Get Lesson Code. And if you click on that, it will download an R script version of all of the code chunks that are contained within this tutorial. So rather than copy and paste every little thing, you should be able to just click on that and download it. I don't know if it's actually responding to my download request right now. Something is acting weird. Well, that's too bad, because I just deleted the version of this I had in my thing before. All right, so we'll see if that comes up and starts the download for me. But we can start talking about what are the important points of this tutorial. So I'm going to take us back to last week here quickly. And what we did last week is we went through the data portal. We learned how to use that. We talked a lot about what are the important pieces of information you need to give to the data portal. You need to specify the data product that you're interested in, the time ranges that you would like to see, if you want to specify down smaller than just all of the data. And you can also specify which sites that you want to collect data from. We downloaded a .zip file from the data portal, and we were looking at PAR data, this photosynthetically active radiation. So it downloaded a zip file. We unzipped that zip file, and it contained quite a bit of stuff within it. So I'm just going to delete that for now and kind of take a fresh look at this. So it had all of these PDFs, these documents that are important, and then another zip file within it containing one site month worth of data. And if we take a look at those data again, within that site month of data, we will have a whole bunch of data files. So these are for the 1 minute and 30-minute averaging intervals. And because we're talking about a PAR sensor, we also get an individual data file from each one of the sensors that is connected to the tower. So again, our towers have multiple sampling heights on them, as indicated here in the sensor positions file. And then each one of those tower positions then has a sensor, which has its own data file, which is saved as a comma-separated values file, a CSV. So if you wanted to actually go in and analyze, data across multiple heights, across months, across sites, it can turn into a total pain. Like, how are you going to deal with all of these CSV files and make it go smoothly? So that's what we're going to talk about today. What I'm going to do now is just actually delete this version of the file so that I'm working just straight from the raw zip file. And I'll take you into R to talk about what we can do to actually work with these files in R. I'm going to try to download this one more time. Maybe I'll refresh this page. Oh, it's blocked. Is that what it is? Okay, that's what the problem is. It's probably going to give me a flood of these now. Cool. All right. So hopefully this works now. Yeah, great. Cool. All right. So I'm in our studio at this point. This is a group talking about R, so you're probably familiar with this environment. I cleared out my global environment and my plots, and I'll clear up my console before we get started. All right. So before we start munging the data, we'll have to pull in some of these packages. In this .r file, we have quite a bit of stuff that's commented out by default. So the install package commands, if you still need to install any of these, you can just remove these hashtags in front of it and remove the comment and install them that way. I'm going to kind of assume folks have already installed those or can just install them up above to catch up with us. And then I'll run these library commands. Let's see, this library, the RHDF5 package is, we're actually not going to need that for today, so feel free to comment that out if you so choose. And the main package I want to talk about today is this Neon Utilities package. So this is one, it is on CRAN. It's developed primarily by Claire Lynch and some of my other colleagues. So we'll talk about the tools that Neon Utilities has to make all this easier for us. I'm going to run this line to set strings as factors to false. I think we discussed before. Oh no, this is in a different environment. But with the new R version 4.0, you shouldn't have to run this line, but it's not going to hurt. So don't worry about it either way. Okay, so what we're going to do now is on this line 28, this is a function coming out of the Neon Utilities package called stack by table. And what this is going to do is take all of those CSV files and combine them into a single file that should be a whole lot easier to work with. To run this command, you're going to need to enter in the location of that par file. If you don't have one on your computer right now, don't worry about it. You can just kind of watch along. If you do have that file on your computer, it may or may not be in the exact same place that I have it. So I want to show you a trick that I learned fairly recently, and it totally changed my world. It's called tab completion. So this should work if you're on a Windows or a Mac. I'm working on a Mac. So instead of typing C drive, I use this little tilde to kind of go to my root directory. And, or if you're on a Windows, you can type, your C drive and a slash. But once you're starting to type in a file path here, if you hit the tab button on your keyboard, it should bring up all of your options for directories and files that are within that current file path that you're typing out there. So I know it's in my downloads folder. I can hit tab again, and I can even start typing in the name of the file. It's neon underscore par, and it'll bring up all of the relevant files in my computer that will match that term. So I'm going to run this stack by table command. And you can see it's doing quite a bit of stuff here in my console. It's thinking, it's doing some things. But what I want to show you is what it's really done to our file. So I had just deleted this folder so we could have a fresh one. But it made a new version of that neon par directory. And instead of all of the additional zip files contained within there for each site month, Now there's just this directory called stacked files. And within there, instead of having different 30-minute averaging CSVs for all of the tower heights and for the different months, now we just have a single table where all of those have been combined into a single location. This will also work if you have multiple sites that you have in your download. Hopefully you can see at this point that this is going to save you a whole lot of time and frustration to bring all these files together, just using that stack by table command. But that stack by table command is really only going to work if you already have this zip file on your computer that you downloaded from the data portal. So I'm going to show you one other function that will download the zip for you and also run this stack by table command and do a couple more things to really quickly bring some Neon data into your working environment. Okay, just looking at my notes, make sure I get everything here. Cool. So because we're talking about R, this is an R group here, I'm going to go into a little more detail about how I like to work in R. One thing I don't trust is a black box. I always want to see what's going on under the hood so I can trust that all of my assumptions are valid for my statistical and scientific analysis. Now to do that, I usually like to look at the functions that I'm running to make sure that they're doing the things that I expect them to do. To do that in R, you can either highlight the function or just copy it and type that into your console. But if you type a function name into your console, it'll bring up all of the source code that is contained within that function. So what I'm going to do is do a Command C to copy all of that. And another one of my favorite tricks in R is if you type Command Shift and the letter N, it will make you a new script that you can populate right away. And then I'm just going to paste right into that new script that I just made. So I like to take a look at this because I want to know what's going on. There's basically a couple things at the top saying that this data product can't be used with this function, but what this load by product function really does is run a function called zips by product, which downloads stuff from our data portal, and then runs this same stack by table function that we were just running in the line above. So now that I've taken a look under the hood, I basically wanted to show you all that because this totally changed the way I work once I could actually figure out what's going on with these functions. But let's take a quick look at the arguments for this load by product function. I'm going to pull up the help file for this function. To do so, you can just type the question mark symbol and then the function name to pull up the help file here. And I like to look at these help files to get a better understanding of what's the expected behavior for this function, and also what are the arguments that go into the function. So argument is kind of a silly word for it in my opinion, but anything that goes inside of these parentheses in this function is called an argument. So these are the pieces of information that the function needs to know to do, what the function does, to run the analysis that you're trying to do. So the reason why I like to go into the data portal first is to understand why we would have all of these arguments in the first place. So this load by product function requires this pretty much the same information as what goes on in the data portal. It wants to know what the data product ID is. It wants to know which site or sites that you're interested in. You can use this C notation to indicate multiple sites if you want to download more than one. You can define a start and end time. You can do the basic or expanded package, much like on the data portal. And then a couple others. But the one I really wanted to point out here is this check.size argument. And what that does is run this function. It's going to call our servers, figure out what's the estimated download size, and then prompt you if you actually want to download that much information. For this data product, this is a vegetation structure data product. It's not a big deal. They're pretty small. So yeah, sure, I'll download a half a megabyte. But why I bring this up is this check.size argument is always going to have a prompt where you need to go in to the console and type a response to download it or not. This is going to crash your script if you're trying to do this in an automated way where you download a bunch of stuff, do some analysis. So if you are planning to use this load by product function in some sort of automated environment, you just want to set this check.size argument to false so that when you run it, it just does it automatically, does it really quickly. Okay, so what does this load by product function do that's different than stack by table? Well, for one, it automatically loads the result into your global environment. So now we have an object called veg_structure that is a named list. It's a list of 7. I'll go into that in a minute. What it doesn't do is load any of this information actually onto your computer and save it at all. So there's no vegetation structure data in my downloads folder. Basically, it downloads all that information, deals with it, loads it to the global environment, and then deletes everything off of your computer that it downloaded. So keep that in mind, because every time you run this function, it's going to pull it down fresh, which has its benefits and its drawbacks. So talking about this named list here, these lines 41, 42 are commented out, but I'm going to uncomment them just to run and see what this looks like. So this particular veg structure object is a named list with 7 list items. You may recognize these as being kind of the various tables that you may download. So the README table, the variables table is there. This is a different type of data product than the PAR data, so our other tables look a little different. You can also view individual tables in there, but I really like this line number 48. This list to end function will basically unlist everything that's in the veg structure object and put it into your global environment. So if you run that line number 48, now you're going to populate your global environment with the various tables that we have in this particular data product. The next couple of lines we added in there but commented out. If you want to save any of these tables to your computer so that you don't have to download them again later, it's not a big deal when they're tiny files, but for other things, it can really be a total pain. So that's just in there for that. And then I'm going to just go pretty quickly through the last couple of things here. Sorry, we don't have all that much time, but I wanted to get you kind of nicely set up to pursue this on your own time if you're interested in that. This line number 65, it's by tile AOP. So again, AOP stands for our Airborne Observation Platform. This is our remote sensing data. And what tiles mean is we post-process all of our airborne data into 1 kilometer tiles, so you have nice squares to work with. So that's just one type of data product that's delivered for those. I'm going to run this line as well. Now it checked and says, do you want to download 4 megabytes of information? I hit Y, because sure, why not? So I'll download some data there as well. So what we just did was 3 different methods for getting data from Neon servers onto your computer. We talked about the data portal last time and how to deal with data portal zip files. We talked about the load by product function, which is really my personal favorite, and the by tile AOP function if you're working with AOP data. The next few lines here, lines 70 through 75, will discuss how to work with reading in those tables if you use the data portal and stack by table workflow. So you can read those in there, change the file paths however you need to. But with this read table Neon function, it will need both the data file, which in this case we're working with the 30-minute averaging interval data, and it's also going to need the variables file as an argument. So you enter in the variables file there. And the reason for that is, if you remember before, and I'll pull up this variables file from the vegetation structure data. In the variables file, it discusses every variable that's reported in the various tables that are downloaded. And it also indicates the data type. Now, this is really important for automating the process of kind of importing these data, because for some things, you're going to need to tell R what is the data type that it's reading. Is it a date time string? Is it a regular string? Is it an integer? Is it a float? Things like that. So this read table neon function is going to need that variable's information to appropriately assign all of the variables that it's reading in. Okay, what else here? I'm going to just take a quick look at our schedule. I think I'm already, okay, I have another few minutes here. So try not to leave us short on time at the end here. So what else did I want to get into? I'm going to skip a few lines here. You can plot out some of the par data, which I think is great. There's some, if you're not familiar with this which function, I would absolutely encourage you to go in and play around with that. I think it's a great way to take a look at our various data. I want to go down here to line 102, because this is going to introduce you to a GeoNeon function. So GeoNeon is another package related to neon data. This one is still currently in development, and I understand some functionality was very recently broken because of an update to some dependencies for this package. So if it doesn't work for you, don't be too alarmed. If it's something important, send us a message and we'll try to get you back on track. But Claire is currently in the process of fixing this package right now. But I bring this up because this GeoNeon function is really important for getting highly precise XY locations for some of these observations. So in this vegetation structure data set, which we're going to analyze using this function, first we'll run this line 104, which will give us the names of all of the variables contained within the mapping and tagging table. So that table contains information for like what plot is this measurement taken in, stem distance, etc. Something I guess I should back up. This vegetation structure data set are measurements of trees out there in a forest. So these trees are identified to species when possible. They have indications if they're healthy or if they're dead. And they also have an XY location. But the way that we measure that XY location is not precise, like to the meter, the way that we deliver these data originally. So if you want to get a kind of a much higher precision on exactly where these trees are, you're going to need to go through this process here, which is the get location TOS function. Okay, so this function will need a couple pieces of information. It's going to need this mapping and tagging table, which again has information like, the scientific name for the tree, where is the XY? I'm not seeing it here. Just other, I guess, important information about that tree. And what this function will do is it will take, I believe it's the UID, the unique code for each one of these trees, and it'll send that to our Neon servers. And then the Neon servers will return the highly precise XY location that's already been kind of post-processed here on our Neon servers. So if you run this line 105, it usually takes quite a while. I have a very fast internet connection where I am here. So we'll probably runs a lot faster for me. But if you run this line 106, you'll see that the names of this table here, the various variables that are reported in this table, are now longer than they were before. We had 29 previously, and now we have 37. So there's additional 8 names that have been added here are things like the UTM zone of the observation. And then the adjusted easting and the adjusted northing, those are going to be your much higher precision XY locations for each one of these trees that are in there. We also have a decimal latitude, longitude, if you prefer to work in those formats. And we also indicate kind of what is our XY precision with this coordinate uncertainty. So I'm just going to run a couple more lines here quickly. This line 112 will merge a couple of those tables together to give you some additional information. And then I like this, this line 119. It's a symbols function. So it will actually plot out these trees. And what we've done here is we're plotting the XY location using this adjusted easting and adjusted northing variables that were just added to this data set. But then we're also using this circles function to give us kind of a 2-scale version of what's the diameter of these trees. So this plot that we end up with here is kind of a 2-scale version of exactly what trees are there in this plot, how big are those trees. And I think this is a nice starting off point, because if you're trying to understand what these forests look like, it's nice to know the physical structure. And then there's also things you could do, like changing the color of each one of these trees depending on the species type and things like that. So last thing I'm going to point out here is how to work with the AOP data. So we downloaded an AOP tile earlier, and it is a TIFF, a geotiff, which you can see in the file name in this line 128. But this file name, I think, is actually kind of obnoxiously long. So what that by tile AOP function does is it downloads that tile, but it also puts it into a very thoroughly nested data structure where it'll save it to your downloads folder or wherever you default to download things. And then it'll save the data product ID and then the year in a separate directory, and then full site in a separate directory, the domain in another subdirectory, and then the actual site in a subdirectory of that. And then this is the level 3 product in a subdirectory of that, et cetera, before you can finally get down to where the TIFF is actually located. The reason we do that is it's annoying if you just have one file, but if you download like 30 or 150 or 10,000 of our AOP tiles, this actually becomes extremely convenient, because this by tile AOP function will automatically organize all of your AOP data into these nested files to keep them separated, so you can find exactly the site or the year or the data product that you're interested in after you download this whole flood of AOP data. So if you're familiar with working with rasters, you'll know what this raster function is all about. And then you can just plot that raster here, which is fairly straightforward. What we have in this image, this is from the Wind River Experimental Forest. These colors are indicating how tall the trees are. So one reason we really like this tile for examples is we have some extremely tall trees, above 60 meters tall in height. But then we also have this really conspicuous kind of triangle-shaped clear cut that WREF is indeed an experimental forest, so they do controlled manipulations of the forest by making clear cuts and things like that. So we find that this tile actually has a whole bunch of information and various kind of ecosystem types just within this 1 square kilometer. So if you are just going to experiment around with some of our AOP data, I would suggest that this tile is an awesome place to start. This is the one I use whenever I'm tinkering around with things. And hopefully now you have a better understanding of how to download it and where it will be saved to. So I know that was a cannonball run through this information. There's a lot to cover here, but again, if you would like to review this, please go back and take a look at this tutorial that's listed here. There's all sorts of great information between the code chunks that will explain the different steps that we did. And of course, we're always happy to hear from scientists out there in the wild. So if you have questions or follow-up, please let us know. Happy to discuss that. And if Marie, Arlene, or myself can't answer your question, we will direct it to one of our expert scientists who will absolutely be able to get down to the bottom of it for you. So that is my, my quick overview of the R coding section of things. Let's see if there are any questions that have come in. I don't see anything in the chat window, but yeah, let's just kind of open it up to questions at this point before we move on to the next step.
Audrey, did that answer your questions about how to integrate all the data that you are going to be working with?
Oh yes, it was lovely. And I like the
Perfect.
fact that A, it's being recorded, and B, it's all on the website. So the pieces
Mhm.
that I missed, I can go back and look at the website. I think that's one of the really wonderful things about Neon is you guys have made these tutorials, and they're straightforward, and like you said, I can do it on my own time. So, but hearing it from the expert is always nice as well.
Great. Well, yeah, thank you, Audrey. Thank you for that. And yeah, again, I apologize. It's so hard to get great information across in 25 minutes, but do what we can and hopefully at least spark your curiosity about these things.
Is there anywhere that let's say someone, like brings a data set to some intermediate point that is potentially useful at that point? Do you share, or is there a way that researchers then share those already processed data sets, or do you recommend that everyone kind of start from ground zero to look at a question individually?
That's a really good question. Yeah. Sometimes I think there is a process to kind of make a new formal data set if we really think it's going to be broadly applicable. What I would point people to at this juncture would be under resources, there is a whole section for code resources over here on the very left. And in our code resources, I know this is something Arlene oriented us to last week, but this tier 1 of community contributed code is where we host, it's usually like a link to a GitHub repo, but this is where people who have their own algorithms to make these intermediate or additional data products would share that information. So you can filter to tier 1 specifically and see all sorts of stuff here. So there's a whole package on calculating tree crown area that I believe integrates some of the LiDAR and hyperspectral data to find individual trees and measure that crown area from there. This Neon HS is a nice one for working with our hyperspectral data, which are pretty complicated. Yeah, good question.
Thank you.
All right, I think I'm shorting us here on our very last section, but we wanted to discuss kind of research questions and maybe try to get people spinning the gears on how might this be useful for your own research or in your own world. And I know this is something Marie has thought a lot about. Can I kick this over to you, or do you have any input here, Marie?
Yeah, you can kick this over to me. So I think we could go one of 3 ways. One, if you guys still have a lot of follow-up questions or like just are really curious about the coding section, I think that's okay if we do that instead. 2, I can provide examples of people, mostly like graduate students and postdocs that have used our data. Or 3, we can talk about, data products you're interested in and, how you might use Neon data. So does anyone have any preference?
My guess is some combination of the last 2 is probably the most useful for the variety of people who might have interest here.
Okay, cool. So I know Arlene went over some of the blog articles that have featured Neon researchers. And I just wanted to quick revisit those because some of the studies are super cool. So I'm just going to share my screen really quick. So one of them, which is actually funny because I went to grade school with this researcher. So it's just, bizarre. Sarah Schooler is a PhD student at SUNY and Humboldt State. She kind of has 2 separate advisors. And so what she was looking at was essentially like, can we predict small mammal diversity using our LiDAR data? And this kind of relates to the tutorial that Donald just went over because it uses the LiDAR data that's looking at vegetation structure, canopy height, and tries to look at, okay, how diverse is and dense is our vegetation structure and forests and she did this up in D5, domain 5, which is our Great Lakes domain in northern Wisconsin. And how does that relate to the diversity of small mammals that we see in those sites? And, it relates exactly kind of how past studies have predicted that, more dense, more diverse vegetation structure, more diversity in small mammals. And how she got started in this, she was a neon field technician, and she just started getting really interested in using our data. And it was just before she went to grad school. And that's kind of why she decided to lump this into her grad school projects. And I know that this is just like one chapter of her dissertation, and some of her dissertation is doing other stuff with LiDAR, but not using our data. So like her whole dissertation isn't just focused on neon data, it's just a chapter. So that's kind of one way that you could go. And I'll just put this in the chat so you guys can look at it on your own time if you're interested. There's also a blog article on it that kind of, a little bit that's geared more towards the public, but I would say, read the paper. I think this is super cool because I don't really think of dew. And for my thesis, I looked at effect of drought on high alpine plants species and demography. And so what this person is looking at dew frequency using our flux tower data. And eventually they want to look at how that might affect how that relates to drought and how that affects plant biology and plant populations. And so they just they looked at there's all these different parts. So I don't know much about dew frequency. So I thought this was kind of interesting that like how dew forms, there's a lot of different factors. So temperature, relative humidity, there was one more that I'm forgetting. But they were able to use all of our sensor data across the United States to look at differences in dew frequency. And essentially they found that like, it's very, it changes quite a bit in grasslands compared to forests, which I thought was interesting. And so in the future, they're going to kind of expand on this study and look at droughts associated with dew frequency. And this was, I think, a postdoc instead of a PhD student. I know they are at the Department of Earth and Environmental Sciences in the University of Illinois at Chicago. And then this is not done by a PhD student, but I really like this paper published in Ecography. It looks at how differences in rodent body size, so they used our small mammal data again, predict species richness across a temperature gradient. And so essentially, they found that these differences in traits of body size did not overlap as much in higher latitudes, which makes sense. Like follows that species gradient where like higher latitudes have lower diversity and lower latitudes near the tropics, more diversity. And they also found that trait diversity essentially gets smaller and smaller the higher you go up in small mammals compared to our lower latitudes. So those are just some examples of some data that's been used, neon data that's been used and completely already published. And I'm positive that for the dew example, he's going to go further with neon data and do some more studies. So yeah, if people have questions about that or curious about how to integrate neon data in their research, let me know. I know I just talked for a while and we're running a little bit low on time. So I think just some final questions, if anybody has any for me or Donald, would be great. I'll stop sharing.
I think if any of you, especially if you are like, oh, do you have data on this or, or what, I think those type of questions would be awesome. So I think there's just a few of you left on here, but any of you? Well, we'll definitely share your contact information so folks can ask questions, and we'll share the recording of this too, so people can listen to it. I feel like this is really helpful to see how you can integrate the data and how easy it is to just copy R code off, or just download the R code to do that. So that's super useful. Audrey,
Awesome.
do you have any?
Thank you all so much for inviting us here and taking the time to engage with us. This is always fun for our side of things. I hope it was useful for you as well.
Definitely. I wanted to thank you for all of your time. I know that you put a lot of time into prepping this and coming up with a website for us specifically, and the presentations were extremely well done and informative. And so thank you very, very much to you and all of your team that helped prepare this. I really appreciate it. And I know a lot of our audience members did as well. So thank you.
Yeah, absolutely. Happy to help.
That's really great to hear. And, we're just really excited about scientists using our data and being interested in our data. And I just want to echo Donald's sentiment. If you have questions about downloading the data, or just, what products do you have, feel free to reach out. That's what I'm here for.
Well, thanks so much. I really appreciate it.
Awesome. Yeah, thanks for the time, and have a good afternoon, and happy Friday, everybody.
Happy Friday.
Happy Friday.
Thanks, everyone.
Yeah,
Bye.
thank you. Bye.
Thank you. Thanks, Donald. Bye.